everyone, TGIF. It's another Friday. How has your week been? Well, I hope that you had a nice week and I welcome you to this weekend. Happy Good Friday and Happy Easter in advance to all Christian faithfuls, to my listeners. Today, I'll be talking to you about another drug. It is the Drug Discovery Series, another episode of it, okay? And I'll be talking about the penicillin story. Who doesn't know about penicillins? Yes, I know that. A lot of people know that there is a drug called penicillin, but also it is a class of drugs. Penicillins are broad-spectrum antibiotics. That is, they treat a wide range of bacterial infections like strep throat, ear infections, urinary tract infections. They work by attaching to and damaging the cell walls of bacteria, and they come in various forms. If you are not familiar with penicillin, at least you've taken amoxicillin one time or the other. You're taking ampiclox, which is a combination of ampicillin and cloxacillin. There is the procaine penicillin, you know, that injection powder that some people pour on wounds, others dilute to take injection. Yes, these are the penicillins I'm talking about. And these drugs have saved lots, millions, if not billions of lives. Literally, I'll be taking you through the whole story from the beginning till the introduction of these wonder drugs to the market. So let me get right into it. Who discovered penicillin? Of course, a lot of people know this. Even from basic biology, we know that the discovery of penicillin was credited to Dr. Alexander Fleming, who was a Scottish bacteriologist at St. Mary's Hospital, London. This was the person who actually discovered the Penicillium notatum fungi, which was the basis for the production of the drug penicillin. From this single penicillin, more synthetic ways to produce more penicillin were devised, and even newer drugs in the penicillin class were produced synthetically. But what was the story behind it? So this accidental discovery happened on a fateful morning of September 28, 1928, when Dr. Alexander Fleming returned from a holiday to find a mold growing on a petri dish of Staphylococcus bacteria, which he was studying previously. He noticed the mold seemed to be preventing the bacteria around it from growing, and he soon identified that the mold produced self-defense chemical that could kill bacteria, which he subsequently named penicillin. As Dr. Fleming famously wrote about the red letter date, quote, "When I woke up just after dawn on September 28, 1928, I certainly didn't plan to revolutionize all medicine by discovering the world's first antibiotic or bacteria killer. But I guess that was exactly what I did." Dr. Fleming continued in this pursuit by publishing his findings in 1929. But the scientific community at that time initially showed little interest in pursuing penicillin due to its instability and difficulty to isolate. He continued his research, but due to the lack of enthusiasm and support by the scientific community, he stopped this research. That is, he abandoned it by the year 1931. Actually, Fleming had neither the laboratory resources at St. Mary's nor the chemistry background to take the next giant steps of isolating the active ingredient of the penicillium mold juice, purifying it, figuring out which germs it was effective against, and how to use it. Some scientists even said that the production of penicillin for therapeutic purposes was almost impossible. So, Dr. Alexander Fleming did not see any reason why he should keep pursuing this and offered the mold strain to others. But nearly 10 years later, in 1937, while investigating microorganisms and the substances they produced, two scientists, Howard Florey and Ernst Chain, uncovered Fleming's research. This landmark work began in 1938 when Florey, who had long been interested in the ways that bacteria and mold naturally kill each other, came across Fleming's paper on the penicillium mold. While leafing through some back issues of the British Journal of Experimental Pathology and assembled a team of scientists to work solely on the penicillin project. These scientists were researchers at the Sir William Dunn School of Pathology at Oxford University. Soon after, Florey and his colleagues assembled in his well-stocked laboratory. They decided to unravel the science beneath what Fleming called penicillium's antibacterial action. Of course, there were personality clashes between this team on how to carry out the research. There were disagreements within the labs. There were complexities from how to purify penicillin to how to produce it in large quantity. But after 3 years of trials and errors, they developed a successful but painfully inefficient process that produced pure penicillin. The team finally had enough penicillin to start animal trials. In 1940, 8 mice were infected with a deadly bacteria called streptococci. Only the 4 given penicillin survived. And the paper they published detailing the experiment gained immediate interest, but the team was still severely limited by production. To produce as little as few milligrams of penicillin, they had to produce gallons of mold broth. Therefore, the team resorted to using any available vessel to grow this mold, from bedpans to meat churns to food tins, even bathtubs to store the broth. Eventually, they arrived at a special fermentation vessel which were developed specifically to hold the liquid. Now that penicillin has been successfully produced, the Oxford laboratory became the penicillin factory, and 6 women were enlisted to tend to the fermenting broth and farm a few precious milligrams of penicillin from this every week. These women were known as the Penicillin Girls, and they include Ruth Callow, Claire Inayat, Betty Cook, Peggy Gardner, Megan Lancaster, and Patricia McKegney. But the problem remained how to produce enough pure penicillin to treat people. In spite of efforts to increase the yield from the mold cultures, it took 2,000 liters of mold culture fluid to obtain enough pure penicillin to treat a single case of sepsis in a person. In 1941, the consequences of the team's production problems and shortage of penicillin became apparent with the first human trial of penicillin. A 43-year-old policeman called Dr Alexander developed a life-threatening infection from a cut. He initially showed signs of recovery upon administration of penicillin, but the supply of penicillin quickly ran out due to the limitation in the production, and Albert's injury returned, which eventually killed him because he died 5 days after the supply of penicillin ended. The penicillin project team had to turn to somewhat unpleasant measures to augment the little supply of penicillin they were able to get from their production. Around 80% of a dose of penicillin is excreted from our bodies in our urine and can be extracted and recycled. Therefore, Dr. Ethel Florey, a supervisor for the clinical trials, was regularly observed on the pee patrol, cycling to patients to collect their urines in order to harvest back the penicillin they already used. With the growing success, the team approached pharmaceutical companies to manufacture penicillin. However, with the Second World War in full swing, British industry was not capable of developing a new mass production process, so the team started to look elsewhere. In June 1941, Florey decided to take penicillin to the US in the hope of finding a way to scale up production. In Peoria, Illinois, a new team was set up in the Department of Agriculture's Research Laboratory. They utilized their expertise in fermentation and designed a new technique for deep fermentation tanks to make the purification of penicillin as efficient as possible. The lab in Peoria had an abundance of corn-stip liquor, a byproduct of cornstarch. They discovered that when added to the mold broth, the yield of penicillin increased exponentially. The high concentration of sugars, amino acids, and nitrogen provided an excellent environment for mold fermentation. Aware that the fungus Penicillium notatum would never yield enough penicillin to treat people reliably, Florey and Italy searched for a more productive species. Therefore, they started a global search for strains of mold with higher percentages of penicillin. Soil samples were sent in from around the world, but the solution was found closer to home, as Mary Hunt, an assistant at the Peoria lab, found a rotten cantaloupe melon at a local market, and that was covered with a pretty golden mold. The mold produced 6 times more penicillin than Fleming's original strain. The mold turned out to be the fungus Penicillium chrysogeum. Yet, even that species required enhancing with mutation-causing X-rays and filtration, ultimately producing 1,000 times as much penicillin as the first batches from Penicillium notatum. Now, US pharmaceutical companies were initially reluctant to commit to large-scale penicillin production. However, by the end of 1941, the US joined the Second World War and demand for penicillin skyrocketed. In the war, penicillin proved to be very useful. Throughout history, the major killers in wars had been infection rather than battle injuries. In World War I, the death rate from bacterial pneumonia was 18%. In World War II, it fell to less than 1% thanks to penicillin. From January to May in 1942, 400 million units of pure penicillin were manufactured. By 1943, the US had sufficient penicillin stocks to satisfy the demand of the armed forces of the United States, as well as their allies. Around 20 different labs in the US were producing penicillin for the war front. The increasingly obvious value of penicillin in the war effort led the War Production Board in 1943 to take responsibility for increased production of the drug. The WPB investigated more than 175 companies before selecting 21 to participate in a penicillin program under the direction of Albert Elder. In addition to Lilly, Merck, Pfizer, and Squibb, Abbott Laboratories, which has also been among the major producers of clinical supplies of penicillin to mid-1943, was one of the first companies to begin large-scale production. These firms received top priority on construction materials and other supplies necessary to meet the production goals. The WPB controlled the disposition of all of the penicillin produced. By the end of the war, American pharmaceutical companies were producing 650 billion units a month, including Shenley Laboratories, Pfizer, which played one of the biggest roles in mass production of penicillin. In 1946, penicillin became available for the first time in the UK for public use. It transformed medicine worldwide and ushered in the age of antibiotics. The discovery of penicillin, one of the world's first antibiotics, marks a true turning point in human history when doctors finally had a tool that could completely cure the patients of deadly infectious diseases. Fleming did little work on penicillin after his initial observations in 1928 due to lack of interest by fellow researchers and lack of know-how and equipment for mass production. The cooperative effort of American chemists, chemical engineers, microbiologists, mycologists, government agencies, and chemical and pharmaceutical manufacturers were equal to the challenge posed by Howard, Florey, and Norma Italy in 1941. Ironically, beginning in 1941, after news reporters began to cover the early trials of antibiotics on people, it was Fleming who received the glory as the discoverer of penicillin. And this actually annoyed the scientists that worked arduously to produce this fungi in large quantities and ensured its utilization, in person of Florey and his colleagues in the Oxford Penicillin Project group. They were virtually ignored. That problem was partially corrected in 1945 when Fleming, Florey, and Chain, but not Italy, were awarded the Nobel Prize in Physiology or Medicine. In 1990, Oxford made up for the Nobel Committee's oversight by awarding Italy the first honorary doctorate of medicine in its 800-year history. But since then, bacteria have evolved to become resistant to the original penicillin and many other antibiotics. In his acceptance speech, Fleming presciently warned that the overuse of penicillin might lead to bacterial resistance, which is what we have seen today. Most bacteria have found a way to circumvent and overcome the power and activity of penicillin and even other subsequent antibiotics after it. And now we face the problem of antimicrobial resistance. Although scientists are still working to produce more antibiotics from newer classes that could actually kill this bacteria. If I've learned anything from this penicillin story, it is how providence actually sometimes lays solutions in our laps, but then the onus still lies on us to discover it. If Dr. Alexander Fleming had returned from his vacation that morning and had not taken note of that mold that was eating up the staphylococcus bacteria, maybe penicillin would not have been a thing today. He could have just packed the petri dish and thrown it away without that observation. Also, if scientists like Florey and Chain had not gone ahead to build up on what Fleming had done and abandoned due to lack of motivation, of course, penicillin would not be available today. Also, if they had not gone the extra mile to mass produce this with the help of other people, the 6 penicillin women and the laboratory assistant who also noted a penicillin-like mold on a cantaloupe, which actually helped to discover a better species of the penicillin mold that could produce even better yield than the notatum. Or should we talk about pharmaceutical companies that work together to help produce penicillin en masse for treatment of soldiers at war? Penicillin will not be available today. So this story has gone to show that we have to be observant of our environment. We have to collaborate. We have to give honor to whom it is due, and we have to make hay while the sun shines. If there is no way, try another way. If people do not support your work today, do not abandon it. Maybe tomorrow it will just be the next wonder drug. So this is me encouraging researchers, scientists to keep doing what you're doing. That little discovery of yours may not make meaning now, but it might be the foundation upon which others will build to produce something spectacular that will change lives and save a lot of people in the future. That is it about the penicillin story on today's drug discovery episode of the Anti-Pharmacist podcast. I hope you enjoyed it. I'm trying to improve on my storytelling, okay? So just bear with me and go along. Do not forget to like, to share, to leave your comments. At least now you know how your favorite medication, I say favorite because a lot of people use these penicillins, okay? You know how it came into being in the first place. And this gives you a sense of appreciation that this thing did not just appear out of nowhere. It took years. Look at from 1928 of the discovery till 1945 when it was then appreciated. How many years is that? That is another 17 years before the whole effort came to a head and it yielded fruit. Of course, now you know that penicillin is not only credited to Alexander Fleming. There were other scientists and other people less mentioned that actually contributed to its success. So do not forget to like, to share, to subscribe to this podcast. Also leave your comment. Let me know what you think about this story. Let me know if there are other things you want to learn about medications, or tell me your favorite medication, the ones whose story you want to hear about. I would like to reply you. Also, you can reach out to me via social media. My DMs are open on Facebook, on Instagram, on TikTok, Anti-Pharmacist Podcast, on X, Farm Podcaster, on LinkedIn, I'm there as Bimsola at Debisi Jose. You can send me mail at antifarmacistpodcast@gmail.com. I'll be bringing you collaborations as we go this 5th season of the podcast, so watch out for those, okay? And do not forget to subscribe to my YouTube channel, guys. Subscribe, listen to the episodes, like, comment, do all of that. Share to people so that there will be more visibility for the podcast, helping more people around the world. Please, please, and please engage my content, okay? Encourage me and help this podcast to grow. I trust that you do just that. So I'll see you next time. Till then, stay healthy and God bless you. Bye.